Okay, so I'm going to post I think on the schedule today we're supposed to have a quiz. Is that correct? Okay, so I will post a quiz on Canvas. It will be due Monday night. So that'll give you a little time to get caught up. It will cover 13, 14, 15. Okay? Any questions about that?
I love 59.
I'm sorry?
I love 59.
Probably.
How many questions will it be?
I don't know, like the previous quiz.
Okay.
Yes.
How far on Canvas?
No, because I figured out how to get it from TestGen into Canvas, so I can do it all on Canvas now. I'm learning how to do things. I like some things about Mastering Biology quiz better, but
Is there still like a time limit on it?
yes, there'll be a time limit on it. But hopefully Canvas doesn't lag like Mastering Biology. The big problem with giving a test on Mastering Biology is that it has a lot of lag time. And I don't think Canvas does it? I mean, Canvas seems to move smoothly from one thing to the next. So it doesn't seem to have that lag time in it. It will probably come up with the multiple choice questions first, but they'll be scrambled. Not everybody's will be in the same order. And then it'll probably have the short answer at the end. So you can go look at the short answer first if you want to. But you're not going to have time to look up everything. And it is cheating if you do it with another person. I will turn you in for academic dishonesty if you do it with another person. And it's not that hard to figure out. When people get all the same wrong answers, that's pretty obvious. Okay. And it has timestamps on it, so if it's done at the same time, I can tell as well. Okay. Any other questions? So quiz will be put up over the weekend. So make sure you do chapter 14 before you take the quiz, because it will be on the quiz. Okay, chapter 15 is about the origins of life and on Earth, and looking at bacteria a little bit. So if we don't get all the way through 15, that quiz will only cover what we get through. Okay? So this is another view of kind of a timeline that's been in the last couple chapters. So if we look at the origin of the universe and the origin of the solar system and Earth, if you look at the solar system and Earth, if you look at this as a clock face, that's about 1 o'clock. Okay? If you look at the first life on Earth, that's about, 3:15 or something, a little after 3 o'clock. So for a very long time, for at least 1.5 billion years, 2 billion years, there were just prokaryotes, and the Earth did not have oxygen in the atmosphere. We'll talk about how we know that in a minute. Then the prokaryotes made enough molecular oxygen that oxygen appeared in the atmosphere. Then after a period of time, we get some single cell eukaryotes. So that's at about 637. Okay? Then, not until like 930 do we get some multicellular eukaryotes. So for all this time, from about 3 to 9, roughly, you have only single cell organisms on Earth. Then you have the advent of multicellular eukaryotes. That would be mostly like plants, fungus. Then you have animals. Then, and it's not until like 10:30, 11 o'clock that land was colonized. All this time, life was in the oceans, with the water. And then we finally get the colonization of land, and then humans are in this very last little tiny bit. So we've only been on Earth for a very short time. So let's look at some of these episodes. Okay, so this is just saying the same thing in words. The major phyla, the phylum, the animals are divided into phyla, like different kind of roundworms. We looked at the vinegar eels, remember, in lab? Those are roundworms. They're segmented worms, like annelids, like earthworms. There are all kinds of different categories, and we call those big categories phylum. That's the plural of phylum. Okay. So it was about 475 million years ago that plants and fungi finally made it to land, and then we get some land animals. So this is just another picture of this in what we call this gives what we call clades. The way, instead of using the tree metaphor that scientists used to use a lot, now they've switched to this clade mechanism, and it shows major divisions. I forgot to turn on the recording. Stop sharing for a second. Go back to share screen. All right. So this is just another way of looking at the timeline. So the big paradox in life is we don't know how life began. This is called the biogenesis paradox. So bio means living things. Genesis means the beginning, right? So where did life come from? So what the cell theory tells us is that all life comes from pre-existing life. All cells come from pre-existing cells. So if all cells come from pre-existing cells, where did the first cell come from? That's the big question. Okay? If you have to have a cell, then that's problematic. So scientists now think that it's possible that the chemical and physical conditions that were existent on Earth 4.5 billion years ago, or 3.5 billion years ago, were such that it could have generated simple cells spontaneously. And we're going to show you why that is. So this has been developed. This is a 4-stage hypothesis, and I do expect you to know this, the 4-stage hypothesis for the origin of life. Notice that it's called a hypothesis. What does hypothesis mean? What? It has to be testable. What else? There were 2 criteria that hypotheses had to have. They had to be testable, and what else? That's more theory. Now, this is a hypothesis that's at least somewhat accepted. I don't know that I would go as far as to say widely accepted. But what else does a hypothesis need? It has to fit all the facts. Before it's useful, a hypothesis has to be testable, and it has to fit all the facts. Okay? So far, this hypothesis is reasonable. Okay? It's not the only hypothesis out there. It is a hypothesis. Okay? So we look at this scenario, and it seems like the first organisms could have been products of chemical evolution in 4 stages. So this is stage 1. I've got them listed, stage 1, 2, 3, 4. The abiotic synthesis of organic monomers. What does abiotic mean?
Non-living.
Non-living. So the non-living synthesis means dehydration synthesis means we do what? Putting things together, making something, okay? So the non-living making of organic monomers. For example, what were the monomers for carbohydrates? Monosaccharides, okay? And what are the monomers of proteins? Amino acids, okay? So this would be the abiotic making of those simple monomers, okay? And this part of the hypothesis was put forward in actually 1953, so shortly before I was born. So this is Stanley Miller, and his grad student was Harold Urey. And what they did was simulate the conditions believed to be present on Earth before there was any life, okay? The Earth was very unstable, lots of lightning, lots of storms, very hot, and most water was in the form of steam or vapor, water vapor. So he concocted this little system. Now, why do we think that the gases that were present in the early Earth were water, methane, hydrogen gas, and ammonia? That's a good question. That's what my husband would say. He would be like, how could they possibly know what was there 4.5 billion years ago? Well, there is one way, and that is in the rocks, because we know that liquids will dissolve gases. Does anyone ever drink soda pop of any sort, or sparkling water? What are those bubbles? They're carbon dioxide. They're gas dissolved in liquid, okay? So we know that liquids will dissolve gas. Well, all rock starts like from volcanoes. And when a volcano erupts, what's the stuff that comes out of the volcano? Madison, get your mask up. What comes out of the volcano?
Lava.
Lava, and lava is solid, liquid, gas. Liquid, okay? So when the lava comes out, it can dissolve the gases that are in the air. Then what happens to the lava? It gets hard, and it turns into rock, igneous rock. Remember, there's igneous, metamorphic, sedimentary rock, right? Okay, so the gases then get trapped in the rock. And if anybody's ever taken geology, one of the very first experiments they do in geology is drop acid on the rock to see if it's limestone, I think it'll bubble up and stuff, okay? So they can test rock to see what gases are in it. And they can go to different parts of the world and dig down, and they can see what the atmosphere was like at that point in history. Because they can look at rock that develops today from volcanic eruptions. And they know when there's been volcanic eruptions in the past, in human history, and they can look at that rock and they can tell what gases and what proportions those gases are in the rock. So there are ways to tell. Now, I'm not a geologist, I don't know exactly how they do all this, but they can figure out what gases were in the atmosphere. What gas is not there? If we have water, methane, hydrogen, and ammonia, what's not there? Oxygen. Okay? Keep that in mind for a second. Okay? So here we have this kind of semi-closed system. It's not completely closed, but kind of semi-closed system because you're taking stuff out. So we boil water, turn it into water vapor, and then we have a reaction chamber here that has the methane, the ammonia, and the hydrogen gas. And we know that there was a lot of electrical activity. We know there was a lot of lightning. Even today, it's amazing how many lightning strikes hit the Earth every minute or every second somewhere on Earth. And Earth is a lot more stable now than it was 4 billion years ago, because the hotter it is, the more lightning. Think about it in the summertime. When do we get thunderstorms? Generally, it's in the late afternoon when it's hot. So the hotter it is, the more likely you are to get thunderstorms. And the Earth was much hotter. So we have lots of lightning. So that was simulated using electrodes that gave sparks. Okay? So we have our methane, which is natural gas. We have ammonia. We have hydrogen and water vapor. And you put in the sparks. Then you put it through a condenser. What a condenser is, it's like a water jacket. It just cools down the liquid and turns it back into a liquid, cools the gas into a liquid, and then it goes back and it keeps circulating. Okay, like it's almost like it's raining. Cools down enough to rain, and then gets heated back up. And you can take off little samples then for testing. And what they found when they put this system through its paces, they analyzed it. At the end of 1 week, 7 days, Miller observed that as much as 10 to 15% of the carbon was now in the form of organic compounds. So it changed from being in methane to being in organic compounds. 2% of the carbon had formed some of the amino acids that are used to make proteins. That is huge, because you cannot have life until you have proteins. You can't do anything until you have proteins. And boom, within a week, we already have some amino acids. The most important thing that we found, and then I think somewhere it talks about what happened after 2 weeks, but the important thing is that the organic compounds could be made easily under these conditions. Okay? And that's huge. We didn't know that until this little experiment. So, along with lots of other evidence and experiments that came after that, we found that you can get amino acids, you can get nucleic acids or nucleotides, you can get carbohydrates out of the system. Okay. And in a relatively quick time frame. Okay, so that's the first stage. The second stage is the abiotic synthesis of polymers. Okay, abiotic means non-living. Synthesis means putting something together, making something. Of polymers. What polymers have we talked about? Way back in chapter 3, I guess it was. 2 or 3. We had monomers, and then we had, if a monomer is a simple sugar, what's a polymer? Starch or glycogen. Okay. If the monomer is amino acid, what is the polymer? What does amino acids go together to make? Proteins, okay? So we have to be able to make proteins. For this whole thing to work, we have to be able to make proteins. So again, what they looked at was what was early Earth like, physically. What was it like? And one of the models they used was the clay and particular kinds of rock which they think were on the Earth in the early days. In the early Earth, you would have had rain that would be splattering and picking up those monomers from the atmosphere, and those would fall on hot clay, and the water would evaporate very quickly, but it would leave those monomers deposited on the clay. And it turns out that the clay will actually act as an enzyme and help form polymers. Now, again, does it happen fast? No. This happens at a relatively slow rate, and then some of the monomers will go together. Do most of them make things that are nonsense? Of course. 99.9% of it is all going to be nonsensical cooking together of amino acids or of nucleotides or something. But occasionally you're going to get something that could be potentially used for living things. So that's the second stage, is this abiotic synthesis of polymers. Stage 3 is the origin of self-replicating molecules. And 3 and 4 kind of, some textbooks put this before, and some of them will put the next one first. It really doesn't matter. So the origin of self-replicating molecules. So what they have found is that short-chain RNA molecules will assemble and copy each other spontaneously. If you put them in a solution and you get some short chain RNA, and you put in, also in that solution, you put in some loose nucleotides, it will the solution under the right conditions will spontaneously make complementary copies of itself, which is pretty amazing. You stop and really think about that. Okay? And this is another reason we think RNA is more ancient than DNA, because DNA won't do this. DNA monomers don't do this, but RNA monomers will do this. So that's pretty significant. So the origin of self-replicating molecules. So this would be stage 1, this would be stage 2, and then this is stage 3, where it's making complementary copies. And this is just going through it in words. What's happening. Stage 4 is the formation of pre-cells. When we talked about phospholipids, if you remember, phospholipids make up 50% of cell membranes. That kind of gives it its structure. When you put phospholipids in a solution, in a watery solution, they will self-form into little balls, into little spheres. Okay? And if there happen to be some proteins around, they will put those proteins in their little sphere too. And so what we end up seeing is little spheres, and whatever else happens to be in the solution will get enveloped inside the sphere. The sphere itself is mostly phospholipids, but if when it's forming the phospholipid bilayer itself, if there's the right kind of protein sitting around, that might get put in the sphere, and then whatever's in the solution just gets put inside the sphere. So you can pick up and make essentially little cell membranes with some material inside that would be similar to what we would have in a true cell. And then this is just some pictures. This is what a sphere of just lipid would look like, a sphere of protein. You have them both in the solution, you get a sphere that contains both lipid and protein. The spheres can only get so big, and then they will pinch off, kind of like bubbles. If you've ever anybody ever done the big bubbles? We had a contraption when our kids were little that was made of light rope, and you could make really big bubbles. But if you tried to make them too big, they pop. There's a limit. There's a physical limit to how big they can get. So when they get too big, they break off and make smaller spheres. Okay. Then we go from chemical evolution to Darwinian evolution. Over millions of years, natural selection favors the most efficient free cells that can copy themselves, and at some point the first prokaryotic cell evolves. Okay. Stop sharing for a moment. Turn that off for a minute. Okay. So I'm going to tell you how I reconcile all this. Okay? Because when I was sitting in your seat, I was raised in a Southern Baptist conservative household, not fundamentalist, but conservative. And so I had to figure out how to deal with this, okay? So let's think about, we have no life and then at some point, we have life. Okay? So somewhere we go from no life to life. Okay? And there's a variety of ways you can get there. One of the ways you can get there, I was never really hung up on the 6,000 years. That never really was part of my thinking. But if you look at this, it looks like there's one gap. There's a gap between no life and life. Okay? Now, if you take something like this 4-stage hypothesis, you have stage 1, stage 2, stage 3, stage 4, right? Okay? Now instead of having one gap, how many gaps do you have? We have a gap here, we have a gap here, we have a gap here, and then we still have to get to life, right? So we have a gap here. So we've actually made more gaps. Every time we fill in a gap, we create 2 gaps. Okay? Back in the, okay, let's think historically. What ended World War II? What was the event that eventually ended World War II? When was World War II? Okay, come on, I know you guys are not this ignorant. Was it in the 1800s or the 1900s?
The 1900s.
The 1900s. Okay, about when in the 1900s was World War II? I just read something yesterday that said millennials don't know that 6 million Jews were killed during the Holocaust. Did you guys know that?
Yeah.
Okay, don't be like these people who don't know things. When was World War II about? I'm not asking for the exact year, but in the 1940s. Okay? In the 1940s. Pearl Harbor was December 7th, 1941. I am somebody who hates history, okay? And I know this. All right? You guys need to know these things. What ended World War II in 1945? The atomic bombs. Where did we drop atomic bombs? Hiroshima and Nagasaki, which are in what country? Japan. But I thought we were at war with Germany. Well, what?
Japan bombed us first.
I know. This is funny. I'm sorry. Japan did bomb us first. But we ended the war in Europe, starting with D-Day was kind of the end of the war in Europe. So we have a VE Day, which is victory in Europe, and a VJ Day, which is victory in Japan. Okay? So anyway, what ended World War II was the dropping of the atomic bomb. Was that a good thing or a bad thing? We're not going to debate that. Okay? It happened. It is history. It is the reality. Okay? How did we get the atomic bombs? What? Einstein helped. So, science, right? Science helped end World War II, correct?
Yes.
Without science, we wouldn't have, and mostly Germans were better scientists than us, let me tell you. The only reason we won in Europe is because we were really good at blowing up bridges. Okay? We were not the best scientists. Some of the scientists defected to the United States, which was helpful. But they would have had the atomic bomb before us if we hadn't blown up most of the bridges. Okay? But anyway, so science is what helped end World War II in 1945. So from the end of World War II, 1945, until the 70s, 80s, maybe even into the 90s, science kind of was king. Okay? People believed science could fix things. And so there developed a thinking that everywhere that science didn't have the answer, maybe that was what God did. And this is an actual theology. And this theology is called God of the gaps. God of the gaps means we believe in science, although science is not a belief system. We believe in science until science can't answer the question. And everywhere science can't answer the question, we're going to plug in God. Well, if somebody was healed and we can't figure out scientifically why they were healed, then we're going to say God did that. Now, we don't like to put in when somebody's not healed and say God did that. We don't want to do that. We say science failed them or medicine failed them, right? We don't like to say God failed them. But this doesn't even just take place in science. This takes place in all kinds of endeavors in human activity, this God of the gaps. There's a big problem with this theology, and it is considered a heresy. Heresy means not true, basically. So why is this not a good theology to buy into?
We don't know everything about the universe. Like,
We don't know everything what?
we just don't know everything about within science.
We don't know everything in science. And I'd like to say the word yet, but we're never going to know everything in science, correct? So if we can't know everything in science, there's always going to be more gaps. Every time we fill in a gap, we're going to create more gaps. Okay? The problem is if that's where you plug God in, if you say, okay, this is what God did, and then science figures it out, what happened to God? God gets smaller. Is that a good theology? No. Okay? So, the question is, how can you have faith, which surely by now you figured out I have, and still be a scientist and know when people ask me, it drives me crazy, I do not believe in the theory of natural selection. I accept it as scientific principle. Okay? It's not a belief system. I believe in God. I accept scientific principles because they are. Okay? So, how do you reconcile it? It doesn't create conflict within me and a lot of people I know. The way I reconcile it is that in Genesis, and by the way, you know there's at least 2 creation stories in Genesis. There's not just one. There's 2 creation stories in Genesis. But in both of them, God does what to people that's different than he does to any of the other parts of creation?
He makes them from dust.
What?
He makes them from dust.
Makes him from dust. What else?
He makes them in his image.
What?
He makes them in his image.
Makes him in his image.
He makes the woman from man's rib.
He makes the woman from man's rib. What else? That's different.
He gives them like domain over.
He gives them dominion, but what does he do before he does that? He breathes the breath of life into humans. For me, that's adequate. That separates people from all of the rest of creation. Because God breathed the breath of life into men and women. God did not do that to any other creature or any other living thing. So for me, that is adequate. For me, it does not matter if God literally picked up dust and formed a human being, or if that's a story. If evolution, I'm perfectly comfortable with people evolving. But at some point in time, God breathed the breath of life into humans, which separates humans and makes humans unlike any other organism. That's adequate for me. May not be adequate for you, but that's adequate for me. And I don't worry about what our origins are. I think it's, of course, I'm not into genealogy or any of that stuff. I have a cousin on both sides who's really into genealogy, so that's nice because I can say, yes, we are direct descendants of Pocahontas, you know. Okay. That $1, well now $5 will get you a cup of coffee. Starbucks. I don't drink coffee. Doesn't mean a whole lot. But the point is, be careful how you think about stuff. All right, so I gotta go back to the screen sharing. Why it's not. Okay. So now we're ready to talk about produce. We've already talked about viruses. We're going to talk about produce. First of all, produce are everywhere. They are covering your body right this minute. They are inside your body right this minute. They had dominion over the earth for 2 billion years. And it's just the little trivia here. The number of bacteria in one mouth is greater than the total number of people who ever lived. Bacterial fermentation is used to make lots of things, including dairy products and sausage. Yum. And more than half of our biotics come from a single soil bacteria called Streptomyces. Not all bacteria are harmful. We need bacteria. That's really evident today because everything they like to say is a probiotic. That means it's helping your microbes to live. See, they can cause disease. Okay, here we go. The two major branches of prokaryotic evolution are bacteria and archaea. These used to be in one kingdom that we called Monera for a long, long time. And then two things happened to cause us to actually divide them into two domains. Domains are bigger than kingdoms, like a higher level of hierarchy. And those two things were, first of all, we could get deep in the ocean. Why couldn't we get deep in the ocean 100 years ago? What happens to people if they go deep in the ocean? You die because the pressure is so much. Okay, so we had to overcome the technical difficulty of getting to the bottom of the ocean. And secondly, the thing that happened was molecular biology. So those two things put together created the need for new classification systems. And so now we divide prokaryotes into bacteria and archaea. Most prokaryotes are bacteria, but archaea are a fundamentally different cell type, and that's why they got their own domain. Okay, instead of being a little piece of Monera, they're now their own domain. And they are actually more similar to eukaryotic cells than bacteria are. In ways that this is true, and you do not have to know these specific ways, but they do not have peptidoglycan in their cell walls, which is a chemical that is found in the cell walls of bacteria. They have branched fatty acids. The first amino acid in their polypeptides is methionine, which is the same as eukaryotic cells. But in bacteria, it's something called formal methionine. And they are not killed by most antibiotics because most antibiotics destroy the peptidoglycan. If they don't have peptidoglycan, antibiotics not going to affect them. Okay, so we're going to look at archaea first. It's a smaller group. And two, we're going to divide archaea into three groups. Okay? The first two groups are what we call extremophiles. Okay, think about that word a minute. Extreme means on the edges, okay? All right? And philes means like hydrophilic. What did hydrophilic mean? Loves water or hates water? likes or loves water. So extremophiles like extreme conditions. Okay? So they thrive in very extreme conditions. So one category are called halophiles. Halophiles live in highly salty environments typically. They are not called halophiles because they enjoy playing Halo. They are called halophiles because salt is sodium chloride, and on the periodic table, chloride is in a category that is called halides. Chlorine, bromine, iodine, fluorine, those are the halides. So they're called halophiles because they like salt. And you will find them growing in very salty environments. Now, there's lots of things that live in the ocean, right? So that's certainly not unique. But think about the fact that before there was refrigeration, and before we figured out how to can with high pressure using, pressure canners, what was one of the main ways we preserved food? Salt. That's why we have country ham. That's why we have corned beef. That's why we have things that are very, very highly salty. Is because salt discourages the growth of bacteria, things that spoil those foods. Okay? So we're not talking salt water, we're not talking ocean water, we're talking really salty. Okay? So these extreme halophiles thrive in salty water. Mostly they are aerobic and heterotrophic. What does that mean? Aerobic means they use oxygen, and heterotrophic means hetero means it eats other things. It depends on other organisms or other stuff to eat. It doesn't create its own food. If you've ever flown over the ocean and seen pink patches, that's halophiles. But if there's not enough oxygen in the atmosphere, in the area where they're living, they will flip and they will do photosynthesis and they will become anaerobic autotrophs. So they can actually change their whole structure of getting food. So that's pretty amazing. Some archaea are extreme thermophiles. What's thermophile going to mean? Thermo means heat. So thermophiles are organisms that love heat. So these are found in very hot water. They are found in hot springs and in very acidic soil. And some of these that are free living will make white fluffy balls in the ocean. The ones that live near the hydrothermal vents in the ocean, okay, the hydrothermal vents are places where there would be volcanoes if it was on land, or at least have some activity. And they're releasing hot gases, very hot. Think how hot a volcano is. A volcano is hot enough to melt rock. You're talking extreme heat from the center of the earth. So these hydrothermal vents in the ocean are extremely hot, and they release a lot of hydrogen sulfide. So these organisms that live near the hydrothermal vents can actually do chemosynthesis. Instead of using light as their energy source, they can actually use hydrogen sulfide and the heat to do chemosynthesis and make their own food. So it's pretty amazing. It's a whole little ecosystem down there. And I say little, but think how big the oceans are. It's little because we don't know about it. We don't know a lot about it. So we tend to think it's very small. They live in symbiosis with some really huge worms on the bottom of the ocean. If you are interested, you can Google pictures, and it's pretty bizarre. Just two worms that are many feet long, but they're the bottom of the food chain. A third category of archaea are the methanogens. Okay, methane is what? It's a gas. It's natural gas, regular old natural gas. Gens. What does the term gen sometimes mean? It can mean different things. It can mean generation, like parents' generation, your generation. What else can it mean? Passages, okay? How about just making something? Generate as in making something. You're generating energy, okay? So what methanogens produce methane. So they are found at the bottom of lakes and swamps. If you ever go somewhere, anybody been to New Orleans or some Louisiana where there's lots of swamps and stuff, there'll be bubbles of gas coming up. And those bubbles of gas are generally methane. Which, by the way, has no smell. The reason you can smell natural gas is because they put nasty odor in it. They put something in there so you can smell it so that you know if there's a leak. Otherwise, you wouldn't know there was a leak. It's also found in the digestive tract of lots of animals, including us, because we have bacteria in our, we have protists in our, prokaryotes in our digestive tract that produce methane. If we could harness that methane, we would have a never-ending source of energy. Now, this wouldn't completely do away with the whole greenhouse effect because you still get CO2, but you don't get all the pollution that goes along with extracting methane from the ground. The reason oil prices have gone down so much and gasoline prices and stuff have gone down so much in the past 10 years is because of fracking. Fracking is where they inject high pressure water in rock to extract mostly methane. Some fuel oil, you get some oils too, but largely to get methane. What's the problem with fracking? What? It's really bad for the environment. Why? It's not, some people say it's not as bad as mining coal, but you're going to extract lots of oils and stuff that are going to contaminate your groundwater because you're forcing that stuff out. It increases seismic activity. What happens if you increase seismic activity? You can have earthquakes and stuff. So, you know, go in there and start messing with rock, you're potentially creating problems. But if we could harness this in a cost-effective way, you could take your waste, you could take your dumps, your landfills, and figure out a way to extract the methane, and you would have basically all the energy you need. On the bad side, it does contribute to greenhouse gas because methane is also a greenhouse gas. All right, now moving from archaea to bacteria. Okay, we're still in prokaryotes, but we're leaving archaea and we're going to talk about bacteria. What we typically think of as bacteria. So we looked at the structure a little bit back in chapter 4. We know that prokaryotic cells lack a nucleus. They also lack other membrane-bound organelles, such as mitochondria, chloroplasts, and vesicles, stuff like that. Prokaryotes have a cell wall that is exterior to their plasma membrane, but it's not made of cellulose. Plants, their cell walls are made of cellulose. In prokaryotes, in bacteria, the cell wall is generally made of peptidoglycan. And we say that prokaryotes reproduce by something called binary fission, not mitosis, because mitosis means dividing of the nucleus. And if they don't have a nucleus, they can't do mitosis. So they do binary fission. Under the right conditions, and your body happens to be in pretty right conditions most of the time, bacteria can reproduce extremely fast, usually 20 minutes. You can go from 1 bacteria to 2 in 20 minutes, to 4 in 40 minutes, to 8 in an hour, which doesn't seem like very much until you start doing that every hour. And within 24 hours, you have way over 1 million bacteria. So pretty fast. Not as fast as viruses, pretty fast. Most prokaryotes are unicellular and very small. We looked at Anabaena. Remember we looked at Anabaena under the microscope. That looks kind of like this. And that is a prokaryote. Occasionally, some prokaryotes form true colonies and show a little bit of specialization of cells and are large, but most are not. Now, one of the things that I think is really interesting about prokaryotes is that a few prokaryotes form what we call endospores. Under harsh conditions, some bacteria will basically take the genetic material of a bacteria and encase it in a hard shell and discard pretty much everything else. Don't need a whole lot of ribosomes, don't need a whole lot of cytosol, don't need much else. And they form kind of a hard case. So you've basically got a very small bacteria inside a hard case. And these endospores can survive very harsh conditions, including living in a jar of green beans. Okay? And the endospores find this wonderful. The endospores of botulism. And they grow in they like to grow where there's no oxygen, so in anaerobic conditions. And what will happen is if there's endospores everywhere, and under the right conditions, then they will grow. So in the olden days, people used to get botulism from green beans frequently. And the way you could tell was if the can had a bulge in it. You know, if you've ever done any canning, the canning lid is supposed to go down. If there's bacteria in there growing, it will produce gas, which will cause the little dome lid to pop up instead of staying down. When that happens, what are you supposed to do with the jar of green beans? Throw it away, okay? Now, not everybody does that. And if you don't pressure can the green beans, if you just get them hot or boil them in water, you don't destroy the endospores. You have to get endospores to like 240 degrees Fahrenheit for at least 10 or 15 minutes to kill them. And no matter how hard you boil water, it boils at 212 degrees Fahrenheit. So in a pressure cooker, it raises the boiling point of water. As you increase the pressure, you increase the boiling point of water, you can get it hot enough to kill the endospores. They have found endospores of Clostridium is the genus that has forms a lot of endospores in like the tombs, the Egyptian tombs, and then when they're exposed to ideal conditions, boom, they start growing. So they've been in the tombs for however many thousand years, I don't know, that they've been in there, and then those spores can grow once they're given proper conditions. So botulism creates spores, tetanus creates spores. It's called Clostridium tetani. And anthrax creates spores. And that's what makes anthrax so dangerous. You can send it in the mail, which they did right after 9/11, and you can inhale it. It's put in a way so that pressure is released. You can inhale it and it can kill you. Anthrax is everywhere. If it's taken orally, it usually doesn't kill you, but if you breathe it, it does. So we can sort bacteria lots of different ways. Some are aerobic, some are anaerobic. Pseudomonas causes urinary tract infections. Legionella causes Legionnaire's disease. Anybody familiar with Legionnaire's disease? It's probably the closest in some ways to the pandemic we're in now. There was a American Legion convention, and all these people came down with this really severe pneumonia, some of whom died from it, and they couldn't figure out what was going on. And it turns out that Legionella pneumophila goes through HVAC systems. Now, as far as I know, they haven't really seen that with the coronavirus. But it took a while to figure out this Legionnaire's disease because they didn't realize it was going through the HVAC system. Staph aureus causes staph infections, but interestingly, again, it will shift to anaerobic. If it's in a low oxygen environment, boom, it can switch and do anaerobic. Anaerobic bacteria include salmonella, which causes food poisonings, often found in chicken, some in eggs. E. coli causes food poisoning, Clostridium botulinum, and Clostridium tetani. By the way, you can find Clostridium botulinum in honey, raw honey. You should never give raw honey to an infant. You should never ever sweeten their food with honey. And what happens is our digestive system is such that if we take in honey and the small amount of botulism it might have, the acid in our stomach will destroy it and we're fine. But infants don't have as much acid in their stomach and the right digestive enzymes. And so if they are exposed to even a small amount of Clostridium botulinum, they can die from it. And it was parents who were trying to give their babies the very best thing that were sweetening their cereal with raw honey from the local food, farmer's market kind of thing. And some babies died from it. So never ever give an infant raw honey. Aerotolerant means doesn't bother, they don't really care if there's oxygen or not. So streptococcus causes most strep infections, is aerotolerant. Bacteria are separated into categories by their shape. A cocci is like a little sphere. A bacillus is what we call a rod. And spirochetes look like this. They're strange little shapes. They look kind of spiral. The organism that causes syphilis is a spirochete. About half of bacteria have flagella. They can be gram-positive or gram-negative. That's just a stain that's put on them to see if they take up the stain or not. The ones that are dark purple here are gram-positive, and the ones that are kind of pink there, that's gram-negative. That matters because different antibiotics work on gram-positive and gram-negative bacteria. And so if you've heard of broad-spectrum antibiotics, they work on both. But a lot of antibiotics only do work on gram-positive bacteria. Some other ways to sort bacteria include using nutritional diversity. And all this means is you can have photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. Most are either chemoheterotrophs, which is what we are, or photoautotrophs, which are organisms that do photosynthesis. These are less common. A lot of heterotrophs end up being decomposers. The ones that are not photosynthetic or chemosynthetic, a lot of them are decomposers. Lactobacillus, lacto meaning milk. Lactobacillus is used to make cheese, sour cream, yogurt, buttermilk, things like that, cultured products. A lot of them provide antibiotics. For example, this one, I'm not going to try to say it, makes erythromycin. They produce vitamins. In your digestive tract, there are E. coli. E. coli makes vitamin K. Newborns have a sterile digestive tract. They don't have any E. coli in the digestive tract yet. So they don't make vitamin K. Does anyone know what vitamin K does for you? What's important about vitamin K? It's really important for blood clotting. So almost always they give a newborn an injection of vitamin K because if the child has any kind of issue and he or she bleeds, they can bleed to death very easily because they don't have any vitamin K in their system. Babies that nurse get vitamin K quicker than babies who are fed formula, simply because they pick it up from the mother. It turns out that photosynthetic bacteria are the source of much of the Earth's oxygen. Okay, I told you I'd come back to this. I think I told you that. The early Earth had no oxygen in it. And then after there were photosynthetic bacteria over a period of like millions and billions of years, it built up enough oxygen in the atmosphere. Because frequently people will look at that first experiment, that Miller-Urey experiment, where you were making the abiotic synthesis of monomers, and they will say, if that happened then, why isn't it happening today? Why don't we have the abiotic formation of life today? The reason is there's oxygen in our atmosphere. What would happen today, or in that experiment, if there was oxygen in that chamber? And you have hydrogen, which we know is very highly flammable. Think Hindenburg, the big blimp that blew up. So you got hydrogen. You got methane, which is natural gas, which is flammable. And you put oxygen in there, and then you add a spark. What's going to happen? Boom, it's going to blow up your equipment. Okay? Because the presence of oxygen means that you can have fire. In the absence of oxygen, you don't get fire. Because how do we put out fires? We somehow remove the oxygen supply. We smother it in some way, shape, or form. Okay? So the fact that there was no oxygen in the atmosphere allowed the process to happen. You throw oxygen in the atmosphere, it doesn't happen anymore. It can't happen. So it wasn't until we had enough photosynthetic bacteria that we could have oxygen in the atmosphere. And oscillatoria, which is another similar to the Anabaena that we looked at, is one of the algae that creates oxygen in the atmosphere. Interestingly, some autotrophic bacteria can also fix nitrogen. What that means is it takes nitrogen from the inorganic sources and turns it into organic nitrogen. We breathe air that is approximately 80% nitrogen. But we can't make our own proteins. We can't incorporate that nitrogen in our cells in any way. We breathe nitrogen in, we breathe nitrogen out. Plants do the same thing. They breathe nitrogen in, they breathe nitrogen, well, they don't really breathe, but they take some in and out, okay? But some bacteria can take that nitrogen and turn it into compounds that plants can then turn into amino acids. And without those bacteria, we wouldn't have proteins. So Nitrobacters is a bacteria that fixes nitrogen. And by the term fix, we mean take it from being inorganic to being organic. Bacteria are very complex. They will move toward light of a desired wavelength or move away from light of an undesired wavelength. They will move away from toxins. They have receptors on the membranes. They reproduce by binary fission. And colonies can move as a whole unit. We're not going to go into any details of binary fission. They have one circular chromosome. There's one. And circular not because it's in a circle, but circular in that it's continuous. And they replicate that one chromosome, and then they split into two separate cells. That's called binary fission. So some bacteria that causes diseases we call pathogens. Most bacteria cause diseases and problems because they produce poisons, endotoxins or exotoxins. Exotoxins are proteins that are secreted by the bacterial cells. In other words, they exude them from themselves. And this is found mostly in gram-positive bacteria. Now, I mentioned botulism. Here's what happens with botulism. You have your jar of green beans, and the person, generally in the olden days, the grandmother or mother, opens the jar of green beans. And if it's got botulism, that means there's some pressure. And she may breathe in, because it becomes aerosolized, she may breathe in some of that botulism spores. My mother says my grandmother always tasted a green bean at the top of the jar before she cooked the green beans, heated them up, which is really, really dumb, because if you dump them in the pan and boil them, then you destroy the toxin. So the person who usually died of botulism was the mother or grandmother, whoever opened the jar. So cook the food, get it hot first, that destroys the toxin. Okay? She would taste it to make sure it was good. It's like, but if it's bad, it's going to kill you, you know. That's not a good thing. Clostridium tetani causes tetanus. That's what you get a DPT shot for, or Tdap now they give. Corynebacterium diphtheria. What disease does that prevent? Or what disease does that cause? Look at the last word. You've probably never heard of it. Diphtheria. Diphtheria has made a comeback. You know why? People aren't getting their kids vaccinated. That's the D in the DPT, or the Tdap. It's the D in it. And diphtheria is a disease that actually causes a membrane to form across the airway, across the trachea, and you literally suffocate. Both of my parents lost a sister to diphtheria. There must have been a really bad outbreak in southeastern Kentucky in 1927. Because my dad's parents lost a little girl right when he was born. And then my mother, who was born a year later, her parents lost a little girl the year before she was born. And so there was a bad outbreak in eastern Kentucky. Now we have a vaccine for it. Nobody's ever heard of it. And now we have kids coming down with diphtheria again because people aren't getting their kids vaccinated. Staph aureus is a common staph infection. We all have it on our skin. Strep causes everything from strep throat to scarlet fever to rheumatic fever to necrotizing fasciitis, which is the flesh-eating disease, which can be really bad. So those are exotoxins. Endotoxins are chemical components of the cell walls of certain bacteria. Some of these can actually withstand autoclave temperatures, which is frightening. These are generally gram-negative. Salmonella typhi causes, right now? This is one that actually does occur. What? No. It's typhoid fever, yes. Salmonella, the other kind of salmonella is the one that's in chicken that causes food poisoning. But typhoid fever generally happens when there's been a flood, is the most typical. So it must be spread by fecal matter. Bacteria called Proteus Neisseria meningitis. You probably had to have a vaccine against a couple kinds of meningitis in your life. They were just coming out with those when my kids were teenagers. The best defenses against bacterial diseases are sanitation, antibiotics, and education. Wash your damn hands. I can't believe I was in the bathroom the other day and somebody came out of the stall and walked out of the bathroom without washing their hands. I'm like, seriously? I'm more worried about getting E. coli or something from surfaces. I'm not that worried about getting coronavirus from a surface. That I'm worried about getting from the air. From the surfaces, I'm worried about getting salmonella or E. coli or something because people don't wash their hands. And if you're ever in charge of the world, don't put air dryers in bathrooms. People are like 3 times more likely to wash their hands if there's paper towels. If people come out of the stall and they look and there's no paper towels and all there is hand dryers, they'll walk out of the bathroom. instead of drying your hands. Plus, hand dryers, you're spreading germs all over the room when you wash your hand. Hopefully you wash away most of the stuff, but if there's anything left on your hands, the dryer's sending that all over the room. So what do most of us do? We wash our hands, we put them under the hand dryer for a few seconds, nothing happens, and we come out and we wipe our hands, right? Well, your pants are not probably particularly clean where you've been sitting down on yourself, you know, in a chair. You don't want to do it in the front, so you do it in the back. That's like probably not the most effective thing to do, okay? So we'll stop there. There will be a quiz on Canvas. Hopefully I'll get it up by this afternoon.
What does autoclave mean?
An autoclave is a high pressure, it's like a pressure cooker
Oh.
that we use in the lab to make media and stuff. I'm sorry, I should have said that.
Will 11 and 12 be on the quiz or just 13 through 15?
Just 13 through 15.
Okay.